Hello everyone, I welcome you to a lecture on cellular organelles. So, this is our normal cell, isn't it? So now we are going to see about the smooth endoplasmic reticulum. So the smooth endoplasmic reticulum is this tubular network but without the ribosomes. So those bulges which we see in this tubular network are the ribosomes. When there are no bulges, then it is smooth, and that is known as the smooth endoplasmic reticulum. This is how it looks in a two-dimensional form, and this is how it looks in a three-dimensional way. It is connected to the nuclear envelope. What is the function of the smooth endoplasmic reticulum? The main function is to synthesize steroids as well as lipids. Since the steroids and lipids are most synthesized in the testes as well as in the adrenal glands, they are prominent in the testes and adrenals. The testosterone is a steroid, isn't it? All the hormones that is produced by the adrenals and many more that are produced by the adrenals are lipids. So therefore, the smooth endoplasmic reticulum is prominent in the testes and in the adrenal glands. In a rough endoplasmic reticulum, as I said previously, the one which has the ribosomes attached to it is the rough endoplasmic reticulum. What is the function of a ribosome? The function of ribosome is protein synthesis. Once the ribosomes produces the proteins, it causes exocytosis. There is COP1 and COP2. So what happens is the proteins which are produced from the rough endoplasmic reticulum will get transported to the Golgi apparatus. So when it gets transported from the rough endoplasmic reticulum to the Golgi, that is anterograde transport to the Golgi is by COP2 transport. COP1 transport is the retrograde transport from Golgi to the endoplasmic reticulum. Fine. So how will the questions be asked? The question will be such that there is trafficking of proteins to the Golgi apparatus. So from rough endoplasmic reticulum, the Golgi apparatus, the protein doesn't enter into the Golgi apparatus. In that case, what intracellular change we expect? So if it doesn't reach the Golgi apparatus, then it is staying in the rough endoplasmic reticulum. That's it, right? So there will be dilatation of the rough endoplasmic reticulum since it is retained over there. And similarly, there is another question which in USMLE where you will be asked about cystic fibrosis. They will be giving you a question on cystic fibrosis, and they will ask you, where is the misfolded CFTR channel retained? Mostly we think that it is retained within the cell, isn't it? No, not exactly. It is retained in the rough endoplasmic reticulum. So this is another important. These are the two questions which are generally asked with this concept. Then what are the free ribosomes? Free ribosomes are those which are present just like that in the cytoplasm, and they will be producing proteins for intracellular use. It is essential for cells' internal protein needs. Golgi apparatus. So what is the function of Golgi apparatus? So this is the Golgi apparatus. We need to know that if we have any product to be sent to anyone or any courier to be sent, what do? We generally write the name and the address, and then we pack it in a particular box, isn't it? So that work, that is packaging and addressing and labeling, is done in Golgi apparatus. It processes and packages the proteins. If it writes the right, if it adds the right address, then it will be delivered to the right person. But if it cannot add the right address, the delivery will be impaired. This is what you should know in Golgi apparatus. So the clathrin coated vesicle transports proteins between the Golgi as well as the endosomes. So from the Golgi, the proteins are all produced and they are transported. This is the main defect that happens in the eye cell disease. I will tell you in detail about eye cell disease now. So what happens in eye cell disease is that it is an inherited lysosomal storage defect. So it is a lysosomal storage disorder. What happens? The Golgi is supposed to produce all these digestive enzymes, form it into a small vesicles, and deliver it to the lysosomes. So how will it deliver? It will label it with the right address, isn't it? But there is a defect in this address. That's how I remember this concept. There is a defect in the N-acetyl glucosamine 1 phosphotransferase. So because of this defect, there is a failure of the Golgi to phosphorylate the mannose residues on the glycoproteins. So mannose 6-phosphate has to be phosphorylated on the glycoproteins. So therefore the enzymes will be, so since the mannose is not phosphorylated, the enzymes are just secreted extracellularly rather than getting delivered to the lysosomes. So there is a lysosome, so that in the lysosomes that is deficient in the digestive enzymes. There will be a buildup of cellular debris in the lysosomes. That is the inclusion bodies. Only if the digestive enzymes have reached the lysosomes, then the cellular debris could be digested by these digestive enzymes. Since it doesn't have the digestive enzymes, all the debris stays within the lysosomes. So that is known as the inclusion bodies. And this is the pathophysiology behind the eye cell disease. There is an inherited lysosomal storage disorder. The lysosomes, within the lysosomes, the product is not proper. That is because the label is not proper. What is the label? Label is the N-acetyl glucosamine 1-phosphotransferase. Because of this defect, it cannot phosphorylate mannose residues. Therefore, the enzymes are not getting directed to the lysosomes. Instead, they are secreted extracellularly. This is the basis behind eye cell disease. So, in eye cell disease, it will result in a coarse facial features, gingival hyperplasia, corneal clouding, restricted joint movements, claw hand deformities, kyphoscoliosis, and the plasma levels of lysosomal enzymes is also elevated because they are secreted just like that extracellularly and not within the lysosomes. It is often fatal in the childhood. So this is how the normal lysosomes are, and this is the eye cell disease lysosomes. This is because of the cellular debris, the lysosomes are bloated, bulged, anything we can say, because it is filled with undigested material. What happens to the mitochondria? Mitochondria is the powerhouse of the cell, right? We all know that mitochondria is responsible for the aerobic respiration. So two important events happen there. One is the Krebs cycle and other is the electron transport chain. There is also ATP production in the mitochondria. ATP is the main energy source for cellular processes. Proton gradient is there, which is the key for efficient ATP synthesis. Uncoupling also happens. When uncoupling happens, there is an increased oxygen consumption to the ATP that is produced. Now we are going to see about centrioles and microtubules. So this is a centriole. Centriole is the one which will produce mitotic spindles during M phase. Here you should know that these are the microtubules. They are the key components in this mitotic spindles itself. There will be microtubule inhibitors and microtubule stabilizers. Both are dangerous because if the microtubules are inhibited, then the cell division doesn't happen. Similarly, if the microtubules are stabilized, even then the cell division doesn't happen because the microtubules should be retracted and the cells should be split into two, isn't it? So if they are stabilized, then it cannot be retracted and the cells cannot divide. So what are the microtubule inhibitors? There are 4 important microtubule inhibitors. See, I have drawn the diagrams. This is the cancer cell. So many anti-cancer drugs are microtubule inhibitors such as vinca alkaloids. This is the neutrophils, colchicine, which is an anti-inflammatory, and mebendazole and griseofulvin, anti-helminthic and antifungal. So all these are microtubule inhibitors. On the other hand, we have microtubule stabilizers, which are taxanes, which will stabilize the microtubules, thereby not allowing cell division and resulting in chemotherapy. In addition to this idea, that is the idea of the centrioles and the cell division, can be asked in a different way. I will tell you. So there is a prophase, metaphase, anaphase and telophase during cell division. So during cell division, there is this. So during a cell division, we know very clearly that prophase, metaphase, anaphase, telophase happens. Sometimes they can give you these diagrams alone and ask you which phase does it depend. So at the outset of the prophase, it's very simple. We need not get panic about it. I'll just make it very clear. At the outset of the prophase, the relaxed chromatin, the chromatin is all relaxed initially. They will all be packed into discrete chromosomes, so that during this separation, they does not get entangled during this. So therefore, they all become condensed and they are packed. And so that's what happens in the prophase. In the late prophase, then what happens is that the nuclear envelope begins to break down, facilitating the interaction of these condensed chromosomes with the spindle apparatus. So this is what happens. Once a nuclear envelope, they become vulnerable to the spindle apparatus. Therefore, this happens. The centrosomes, which serve as the, so this is the centrosome. Centrosomes, which serve as the primary microtubule organizing centers, begin to move towards the opposite poles of the cell. This is how it starts. Then this migration is facilitated by the elongation of this microtubules, giving rise to the mitotic spindles, which will capture, align and segregate the chromosomes during this later phase. This entire thing, that is the dissolution of the cellular membrane, centrosomes reaching the poles and formation of the microtubules, all happens during the metaphase. Prophase. Metaphase is the phase where Metaphase is the phase in which all the chromosomes, which are all consisting of two sister chromatids, they will align at the metaphyseal plate. So this is the metaphyseal plate. So a better way to remember metaphase is that they will align at the metaphyseal plate. This alignment is essential to ensure that each daughter cell receives one copy of each chromosome. At this stage, the nuclear envelope is completely dissolved. The spindle fibers are attached to the centromeres of the chromosomes. This is the centromere, isn't it? So here the spindle fibers are attached. This connection plays a crucial role in the segregation of the chromatids during the anaphase. So once this is all well connected, there is segregation of sister chromatids. Anaphase is characterized by the separation of the sister chromatids as they pull to the opposite poles. The centromeric region, which are holding the sister chromatid together, will split, and the chromatids, now individual chromosomes, are rapidly pulled to the opposite poles of the cell by shortening of spindle fibers. In the telophase, which is the final stage of metastasis, once the chromosomes reach their respective poles, they start to decondense. That's what it happens here. Then the nuclear envelope again reforms around each set of chromosomes, and the spindle fibers all break down. So this breakdown doesn't happen during when the microtubules are stabilized, but here during normally it breaks down. And these changes will signal the end of mitosis, and now that it will start splitting into two different cells. This is what happens in the cell cycle. The next cellular organelle is the nucleus. Nucleus is the command center of the cell. We know it is like the headquarters. What we should know is that in the nucleus only there will be DNA replication and there will be RNA transcription. And there is something known as nucleolus. The entire thing is a nucleus, and this is the nucleolus. What happens in nucleolus is the synthesis of ribosomal RNA. rRNA synthesis happens in the nucleolus. If there is a high nuclear cytoplasmic ratio, so the nucleus is very high, that means the DNA replication, RNA replication and ribosomal RNA synthesis, which happens inside the nucleolus, are happening at a rapid rate, right? So if there is a high nuclear cytoplasmic ratio, it will suggest increased mitotic activity. There is one question asked on step 1 USMLE, where they will be giving you about that the nuclear envelope breaks down via protein phosphorylation. So the nuclear envelope, this is the nuclear envelope. So during mitotic change, as we saw on the previous child, then as we saw in the previous slide, the nuclear envelope undergoes dissolution, isn't it? So how will it undergo dissolution? It undergoes dissolution by getting phosphorylated. This is asked in one board exam, USMLE exams. So this is something, a fact that you need to remember. Now we are going to see about the peroxisomes. The key function of the peroxisome is that it will result in the beta oxidation of very long chain fatty acids and fatty acid synthesis too. Similarly, breakdown of branched chain fatty acids and synthesis of branched chain fatty acids too. It will be resulting in Refsum's disease, adrenoleukodystrophy, as well as Zellweger disease. This idea. So we already know that beta oxidation of fatty acid happens in the mitochondria. So what is the role of peroxisomes here? Peroxisome will result in the beta oxidation of very long chain fatty acids. So the fatty acid will be oxidized to a certain level. After that, it can be directed to the mitochondria. So they will be asking you questions such as, a researcher, when he is studying about the cellular organelles, he finds that there is a structure which is small single membraned organelle that contains a crystalline core and a granular matrix. They determine that these organelles are involved in the metabolic processes that causes the breakdown of certain substrates. So this is how the question will be directed, and the subsequent generation of hydrogen peroxide. Now we should also know that because of the fatty acids undergoing beta oxidation, there will be hydrogen peroxide released. So to protect the peroxisomes, there is catalase which is present within the peroxisome which will neutralize the hydrogen peroxide. Which of the following most likely takes place in the structure? And they will be giving you a bunch of questions where it will be very obvious that the one option will be oxidation of fatty acid, or such as say phytanic acid, or any other similar acid can be included in that option. Now let's see about proteasomes. This is how a proteasome will look like. This is how a proteasome will look like. The main function of the proteasome is, once the protein is tagged, it will result in a protein degradation. What tags the protein? Ubiquitination is something which will mark the protein for degradation. Once the protein has a ubiquitin tag, it will be recognized because the proteasome will recognize the ubiquitin tagged proteins, and the proteins will be degraded into smaller peptides. So once the proteins are all degraded, it will be resulting in amino acids, isn't it? The protein will be degraded into amino acids, right? And they can be used for new protein synthesis. This is how it happens. This is a protein and it is ubiquitin tagged. It enters into the proteasome and it comes out such that the proteins are all the protein is all cleaved, resulting in amino acids. There is a concept about autophagic vacuoles in cachexia. The autophagic vacuoles will be increased in muscle cells during cachexia. When does cachexia happen? It happens during starvation and wasting conditions, as well as in cancer, isn't it? So this cachexia is triggered by TNF alpha. The TNF alpha causing cachexia, it will break down the cellular components. With this, we come to the end of our cellular organelle presentation. I have an announcement for you. I'm starting my mentoring lessons for USMLE aspirants as well as for medical graduates. If you like my video and if you want me to mentor you, you can send me a message in the comments and I will get back to you. Looking forward to see you succeed. Have a good day.